Hello, good morning. Let's get started. Today we'll be doing special senses, which is lecture 7. And we will be focusing primarily on the eye as well as the ears. Okay, there is going to be like some stuff here and there as well, but the primary focus is going to be the eyes and the ears. So let's get started. Match the following type of sensory receptors with their functions. Oh, sorry, seems like my pen's not working. Give me a second to try to fix it. Okay, sorry about that. It's working now. Okay, here, first one. We have different types of sensory receptors on the body, and the sensory receptors is going to, be designed to detect a particular type of stimulus. So, for example, a mechanoreceptors is going to be stimulated by changes in pressure or body movement. Okay, so that's going to be right here, A. So pressure would be, for example, if you're poking at the skin, so if you poke it harder, then you're gonna have mechanoreceptors that are located deeper in the skin to detect that. Or if it's just like a gentle stroke on the skin, then you have mechanoreceptors that are on the surface that would detect that. But mechanoreceptors are also going to be found in the inner ear, where when you move your head, for example, sideways, up, down, or like if you're falling, if you're speeding up, accelerating, all that body movement are also going to be detected by mechanoreceptors. Okay, so changes in pressure, e.g. in the skin, you have mechanoreceptors in the skin, and body movement, that would be, for example, in the ear. So that's done. Photoreceptor, okay? Which one is photoreceptor? Photoreceptors are found in the eye, and that is responsible for detecting light energy. Okay, so I'm going to put B right here. There are two types of photoreceptors. There are the cone cells, which detects color, and there is also going to be the rod cells, which are going to be detecting grayscale, and they're going to be most active in during nighttime. Okay, so let me just write that down for you. We'll probably write down again later, but two types of photoreceptors: cone cells, cones, which are for colors, and then we have the rods, which are for night vision. Night vision plus peripheral vision. Okay. Peripheral vision is like your side vision, right? If you see something moving at the corner of your eye, that is the peripheral vision, and that is relayed by rod cells. Okay, next one, nociceptors or nociceptors. That's just another fancy word for pain receptor. Okay, pain receptor. Okay. And they are responsible for detecting damage or oxygen deprivation to tissue. Okay, if there is reduced oxygen supply to a particular organ, for example, it's going to start to hurt, right? And that is detected by the nociceptor. Now, I should also remind you that there are two types of nociceptors. Okay, there is the somatic nociceptor, and then there is also the visceral visceral nociceptor. Okay, the somatic nociceptor is the one that is responsible for relaying primarily damages that occurs on the surface of the body. So if you fall or if you cut your finger, right, all those surface type of damages is going to be detected by the somatic nociceptors. And the visceral nociceptors are the ones that are responsible for detecting like internal damages. Okay, so like if you have a gallstone, for example, and then your gallbladder is hurting, right, that's related to visceral nociceptor. If there is a heart attack and chest hurt, the angina, that chest pain is detected by visceral nociceptors. Okay. What else do we have? Okay, so D is the chemoreceptors, and chemoreceptors are going to be stimulated by changes in chemical concentration. When I say chemical, it's not necessarily like, you know, the stuff that you learn in chemistry class, right? Chemicals can be anything, ranging from things like O2 sensor. You can have like a glucose sensor, right? You can have a sodium sensor. So any kind of molecules, basically, if you change the concentration, your body can detect those changes with the chemoreceptors. And finally, the thermoreceptors, which is quite self-explanatory. It is stimulated by changes in external environment. Does anybody have any question about any of these receptors? Okay, moving on, we're gonna have to identify the different parts of the eyes here. Okay, so first we have attaches the lens to the ciliary body. Let me see if I have like an eye model that I could show you. I don't have a link here, but I assume you have watched the recordings, so I'm just going to fill this in for you. Okay, so attach the lens to the ciliary body. These things, we call them suspensory ligaments. Okay, so the ciliary body, just to give you a quick recap, and we're going to have a chart coming up later that we can fill in, but the ciliary body is essentially a ring of muscle that could contract or relax. When it contracts, it gets smaller. When it relaxes, it gets bigger. Okay, so by contracting or relaxing, the ciliary body is going to change the tension in the suspensory ligament. Okay, and by changing how tight the suspensory ligament are, you can then regulate the shape of the lens. Okay, so just one more time very quickly. Ciliary body is the muscle that could contract or relax, which in turns would change the tightness, the tension in the suspensory ligament. And that change in tension is going to change the shape of the lens. Okay, can someone tell me why it is important to change the shape of the lens? Why do we have to change the shape of the lens at all? No? Okay, changing the shape of the lens is, okay, as Mary said, important for long and short-sighted people, maybe. Not so much, but I think you are on the right track. It's important to change the shape of the lens when we shift our focus from looking at something far away to something that is nearby. Okay, and we'll revisit that concept in a bit. Next question. Fluid in the anterior segment that provides nutrients to the lens and the cornea. So the lens and the cornea, they are transparent structures. They have no blood vessels in them. If there are blood vessels in the lens or cornea, we would be able to see it, right? We will see it in our vision, which of course would interfere with how well we can see. So for that reason, they are going to be transparent, but nevertheless, these are alive structures and they need nourishment. So to provide nourishment, we will need to have the aqueous humor. Aqueous humor. Okay, so that's the liquid that is at the anterior part of the eye. If you look at someone's eye and you see the white part, the white of the eye is the sclera. That is the most superficial layer of the eye, and it is also the location where the muscle of the eyes are going to be attaching to. Okay, you have several muscles that attach to the eyeball that helps it move left, right, up, down, even rotate a little bit. All those muscles attach to the sclera. Next, we have vascular layer of the eye. Okay, so beneath the sclera, immediately, we are going to have the choroid. Okay, choroid. And that is where you will find a majority of the blood vessels, and that helps nourish the 3 layers of the eye, right? And the innermost layer, which is also the layer that contains the rods and the cones. Remember, these are photoreceptors. That layer is called the retina. Okay, retina. So 3 layers of the eyeball. Now, just in case you are a little bit unfamiliar with what I'm talking about, so there is your eyeball, and then there is going to be, like the lens at the front, and then there's the cornea, right? This is, of course, not to scale. Okay. So there's the lens, and this is the cornea, right? So earlier I was saying that at the front, there is some liquid here, right? Okay. That's the aqueous humor. It's the aqueous humor. It nourishes the cornea and the lens. Now at the back, we have another gel-like substance, okay, that fills up the posterior chamber. Okay, so this substance, it helps reinforce the shape of the eyeball, okay, and we call that the vitreous humor. Okay, vitreous humor. Now we don't want the pressure to be too high in the vitreous humor, because at the back here, right, we're gonna have the optic nerve, and if there is too much pressure in the vitreous humor, it's going to be pressing on the optic nerve and damage it. What condition does that lead to? Anybody remember from the lecture? These are questions that could happen on the test, right? So high pressure could lead to, anybody? The condition? Okay, glaucoma. Thank you, Sierra. Okay, so glaucoma, that's right. And that starts with a loss of peripheral vision and eventually blindness. Treatment of glaucoma focuses on reducing the eye pressure. Okay, lastly, the most anterior part of the sclera is going to be the cornea. Okay, which is this convex dome-shaped structure at the front that allows you to gather light from a wide angle. Okay. Next, complete the following chart regarding visual accommodation. Okay, so let me just put a star here and I'll explain to you what visual accommodation means in case you need a refresher. Okay, so visual accommodation refers to the ability to change the thickness of the lens as we shift our focus between nearby and far away object. Okay, there are other things that happens in visual accommodation, but this is the one that we're focusing on. As you, let's say there is, you're waiting at a bus stop, okay? You're reading a magazine while you wait for the bus, okay? And then you hear a bus coming. So you look up and you try to look at the number on the bus to see if it's your bus, right? So you actually have to shift your focus from nearby, reading your book, to far away where the bus is coming towards you, right? So in order for you to maintain focus to see what the letters on the bus, as well as in your magazine, you have to change the thickness of the lens. So in general, you should remember to focus on nearby object, your lens has to be thicker. Okay, thicker. And to focus on far away object, your lens has to be thinner. Okay, so if you look at the lens and the side, it has to be thicker for nearby object and has to be thinner for far away object. This is looking at the lens from the side. Okay, so how do we change the thickness of the lens, right? Okay, so remember your lens, I'm just gonna draw a lens here again. This is side view, okay? Your lens is gonna have these ligaments that are attached to it. Okay, so this is side view, looking at the eye from the side. Okay, if you look at it from the front view, okay, you would see the lens, okay, more or less a circular shape, and surrounding the entire eye is going to be these suspensory ligaments. Okay. Now, stay with me for a second. If all these ligaments pull on all sides, is the lens going to get thicker or thinner? If all the ligaments around the lens pull, right, pull on the lens, will the lens get thicker or thinner? Okay. I hope your choice is thinner, right? Because it makes sense, right? If you're pulling on all direction, then this thing gets stretched out, right? And it's going to be thinner, right? So for that reason, when you're focusing on a far away object, you want the suspensory ligaments to be tightened. Okay. Whereas if you want to focus on nearby object, you want them to be loosened. Okay. Now the ligaments themselves cannot change the tension. The ligaments are just like string. Something has to be pulling on the string. And if you're paying attention, earlier I mentioned there's something called a ciliary body, right? Okay, or ciliary muscle. It's for us, we don't make the distinction. Okay, so this muscle is a ring of circular muscle that goes around the suspensory ligament. Okay, so let me maybe use a different color to show it. Yeah, so there is going to be the muscle, right, that goes around the eye. Now it's important to know that these muscles is a ring of muscle, circular muscle. Okay, so when it contracts, it kind of grabs onto each other and then it makes the circle smaller. Okay, when it contracts, the circle gets smaller. So if the circle gets smaller, then you're not going to be pulling on the ligaments as much. And that means the ligaments are going to be loose and the lens is going to be thicker. So if you want to focus on a nearby object, the ciliary muscle should be contracting. Okay, they contract, grab onto each other, make a smaller ring. Ligaments will be loose, lens will be thick. On the other hand, if you want to look away, if you want to look at far away object, then you need to relax the ciliary muscle so it gets really big, right? But by getting bigger, it actually pulls on the suspensory ligaments, and that in turn spread out the lens because it's being pulled in all direction, and it gets thinner. Okay, so you can almost feel a multiple choice question, right, on the test. Which of the following happens when you focus on a nearby object, right? And then the choices would be contract, tighten, thinner, contract, loosen, thicker, relax, loose, and there's like all different combinations, right? So make sure you know which one is which. Are we together? Does anybody have any question? All right. Let's see. First one we can probably do maybe. Okay, let's do some of these poll questions. Let's see. Go ahead. That's the first one. Oh, sorry. So sorry. It says select all the correct answer. I think I launched the wrong poll here. But you know, let's talk about this. Thank you for choosing the answers. Okay, so mechanoreceptors, right? These are things that would detect changes in pressure as well as body movement, right? So as mentioned earlier, you will find mechanoreceptors in the inner ear. So that includes the cochlea as well as the vestibule. Okay, so if you don't know what those are, we'll review it later when we get to this, get to the ear. Okay, and then we will also find them underneath the skin. Okay, so those are the examples. Okay, next one. Let me launch the right poll this time. Go ahead. Okay, that has been a minute. Anyone else want to lock in their answers? Good. I have 7 of you. Not bad. Okay, so you can see it's kind of all over the place, but visceral nociceptor. Nociceptors are pain receptors, right? And visceral means they're relaying pain that originates from like the inside of the body. Okay, generally things that you don't have voluntary control over. So if you cut your finger, right, that is not from the inside. That is going to be relayed through the somatic nociceptor. So not that. Angina, the chest pain, right? That's from the inside. Kidney stone blocking the urethra. That's from the inside. A bruise from falling, dislocation of the shoulder. Those are all going to be surface type of damage. So only B and C will be detected by visceral nociceptors. Okay. All right, let's get back to it. So next question, we have various eye structure, and we're gonna fill it in. Okay, as much information as we can. Okay, and please stop me if you have a question or you don't understand what I'm writing. Okay, so what is the retina? Okay, the retina, it's pronounced retina. Okay, not retina. Some people that pronounce it that way, but it's retina. Okay, it is the innermost layer. innermost layer of the eye. Okay, the deepest layer of the 3 layers of the eye. Okay, so the retina contains photoreceptors. Okay, cones and rods. Okay, the photoreceptors are mostly located in the posterior half of the retina. Okay, like if you think about the eyeball, right? Here's your eyeball. Here's your lens at the front, like I said. So, you know, no point putting photoreceptors here, right? Because the light comes in, and the light comes in, it's not gonna bounce back and stimulate these locations, right? Okay, so most of the photoreceptors are gonna be concentrated at the posterior half of the retina, okay, where the light is going to be. All right, that is that. Next, we have the fovea. The fovea. What is the fovea? The fovea is a location on the retina that has the highest density of cone cells. Okay. The image of the object of interest is projected onto the fovea, creating a sharp image. Okay. You might remember I mentioned in the lecture, you can only focus on one thing at a time. Okay. For example, if you're looking at the screen right now, you're trying to read these letters, okay? And you know, if you're doing this on a laptop or a computer, okay? If you're doing this on a phone, it's really small, I suppose. But let's say you're doing this on a computer and you're focusing on these letters, you're trying to copy all these words, right? Then you will be aware that you know, there are writings on your keyboard, but you won't be able to see what it is. Okay. So again, right? Doesn't matter if you're doing this on a phone or a computer, I guess. Okay. So I want you to focus over on these purple writings. Okay. Then you are aware that, you know, there is a picture of me on the top right corner, and then I'm talking, I'm doing something, right? So keep reading those letters, right? The ones I wrote. Okay. And then are you aware that I just put up my finger, right? Could you see that at the corner of the eye that I just put up my hand, right? If you keep on reading the letters on the screen, are you able to tell me how many fingers I'm lifting up, right? Probably not, right? Okay, because you're focusing on what the letters are over here. You're focusing on reading these letters. So these letters, these words are projected onto your fovea, right? That creates a sharp image. Everything else that you're not focusing on is going to be your peripheral vision, and we don't have sharp, detailed information from our peripheral vision. Okay, like I said, you would be aware that I put up my hand, but you probably weren't able to tell how many fingers I was holding up. Does that make sense? So you can only have one very sharp, clear image of one thing at a time. Okay, and that is being projected onto the fovea. Okay, next we have ciliary muscle. Okay, ciliary muscle. So we talked about this already. The ciliary muscle is a ring of muscle that controls the tightness of the suspensory ligaments, right? Okay, so it surrounds surrounds the suspensory ligament. Okay. By contracting the suspensory ligaments, will loosen. On the other hand, by relaxing the suspensory ligaments, will tighten, which of course in turn change the thickness of the lens. We talked about that already. Iris. What is the iris? The iris is the color part of the eye. Okay, the pigment that gives the color to the iris is the same pigment that determines our skin color, skin tone, as well as like hair color, for example. Okay, it's the melanin. So the more melanin you have, the darker the iris will be. And you know, you might have a brown eyes, for example, and then the less melanin you have, the iris is going to have a lighter color. Okay, maybe like a blue eye, a green eye, so on and so forth. Okay. But it does more than just give your eyes color. The iris actually helps regulate the size of the pupil. Okay, the pupil is the hole in the middle of the eye that lets the light go in. Okay, you don't want too much light coming in when it's really bright, right? Okay, too much light comes in, then things would appear to be blurry, and it's gonna hurt your eye as well, right? Okay, think about taking a picture with too much light exposure. Right? You can't see anything in the picture, right? So that's why it's important to regulate the size of the pupil. But the actual regulation is, I should maybe put an extra note here. Okay, this is done automatically through the ANS, right? The autonomic nervous system. Remember, we have the parasympathetic system and the sympathetic system. Right? So the sympathetic system is going to dilate the pupil, whereas the parasympathetic system is going to constrict the pupil. Next one, aqueous humor. This is, it fills the anterior chamber of the eye. And the function is to nourish the lens and cornea. All right, next part, we have the vitreous humor, and this time it fills the posterior chamber of the eye, and the function is to reinforce the shape of the eyeball. Okay. Any questions about any of these eye structure before we move on? We all good? Okay, let's see some easy eye question here. Go ahead. Number 4. See if you were paying attention. Okay, I have 5 answers, 6 answers, 4 more. We can do it. Let's see if we can have 100% participation. What controls the size of the pupil? Anyone else? Waiting for 3 more people to answer. Are you guys there? Okay, probably not. The correct answer is the iris, right? Okay, the ciliary muscle controls the tension in the suspensory ligament, so that is not the correct answer. Okay, number 5 should also be fairly straightforward. Please try that. Okay, waiting for one more. Okay, the correct answer is the retina, right? Retina. And you can see here, we wrote it down, right? It contains photoreceptors, right? That's the only layer that has the photoreceptors. Okay, this one is the presbyopia. I don't know. Let's see if you guys remember. Okay, I haven't talked about it today yet, but you know, that was mentioned in the lecture. So let's see if you are able to answer that. Presbyopia. Okay, I have 3 answers, 4 answers now, 4 people, 5 people. Okay, perhaps 2 more. 7 of you that were participating earlier. All right. Okay, let's talk about this. Cannot see far away object clearly. Okay, if you cannot see far away object clearly, then we call that nearsightedness, right? Nearsightedness. Okay, or myopia. You should look at the lecture recordings to see an explanation of what nearsightedness is. Okay, if you cannot see nearby object clearly, okay, that is going to be farsightedness. Okay, the name tells you what you can see. Okay, so in farsightedness, you can see far, but you cannot see nearby. Okay, this is called hyperopia. Okay, cannot see in low light setting. That's like selective blindness. People with low vitamin A, for example, they actually go blind at night. Okay, nocturnal blindness. That's what it's called, right? Because you need the vitamin A for the rod cells to function properly, right? So yeah, that is a lot. Remember last, I don't know if you were with me last semester, we talked about golden rice, right? And that is a genetically modified rice that increase vitamin A level in the grain to grow in places where people have vitamin A deficiency, and that actually helps fix the nocturnal blindness problem. In any case, that's not the answer. Presbyopia happens when visual accommodation is not occurring fast enough. Okay, so those suspensory ligaments, they're like elastic bands, right? As you get older and older, the elasticity in those ligaments starts to decrease, and they are not going to be as responsive as before. So you could be just looking up at something, and then you pick up a business card, or you try to look at your phone, or you try to look at a book, and your lens is supposed to become thicker, but that doesn't happen quick enough or fast enough, and as a result, you're not able to focus. Okay, and that condition is called presbyopia. Okay, so people either get reading glasses, right, or like if you're holding it this close. Oh, you cannot see my book because I'm using a virtual background, but if you hold the book too close, you cannot see. You have to like move it farther away, right? You might have seen older people do that, and that is the answer. That is an example of presbyopia. Does that make sense? Any question about presbyopia and what it is and how to fix it? Remember, to fix this, you need reading glasses. Okay, now this one is complicated. I already explained it in the lecture, but I'll go through it again, okay? Because this is a complicated concept. A lot of people don't get it the first time, so I'm just going to explain it again to you. So we will skip the ear for now, and we're just going to move forward to question number 6. Okay. This is a bird's eye view looking at the eyeballs from the top. Right? So this is the right side. This is the left side. Now, in our vision, we have what's called the right field of view as well as the left field of view. Right? So let's say we put a person here. Okay? This person is on my right field of view. Okay, field of view, FOV. Okay, so obviously on the other side, it's going to be the left field of view. And let me choose a different color here to illustrate this. And this is the left field of view. There's a little blue person here. So which half of the eyeball will the left field of view end up projecting on? Well, light travels in a straight line. So if it enters the eyeball, the left field of view will actually end up stimulating the right half of the eyeball. Okay, so we're going to put here left FOV projects to the right half of both eyeballs, both eyes. Okay, contrastly, the right field of view is going to be projecting on the left half of the eyeball. Okay, the right FOV projects to the left half of both eyes. Does that make sense? This part alone gets people confused sometimes. Anyone have any questions? Yes, Savi, go ahead. No? Oh, okay. All right. So what happens next? Well, let's follow the left field of view and see where it is going to go. Okay. So the left field of view, right, on the left eyeball here, that is going to be transmitted. The information there is going to be transmitted through the optic nerve here. Okay, so there's the optic nerve, and same thing over here, right? The right half of the eyeball is going to be transmitted through the optic nerve. So all these information from the left field of view, right, will be transmitted here. But what happens is, over here, the optic nerve is actually going to continue to travel on the same side. Whereas the optic nerve on the left side will cross the middle and join with the other nerve before continuing. Okay, ultimately, the entire left field will end up going to the right thalamus and then to the right occipital lobe. Okay, so let me just label these structure for you so you know what I'm pointing at. Okay, this is the optic nerve. Okay, left optic nerve, right optic nerve. This becomes the optic tract because once you're in the brain, right, we no longer call it a nerve, we call it a tract. Right? Tract and nerve, they are both bundles of axon. But if you find it in the PNS, we call them nerve. If you find it in the CNS, we call that a tract. And this location where the nerve crossed the middle, we call that the optic chiasma, which is located just in front of the hypothalamus, if you remember from last class. All right, so let's take a look at the other information here. Okay, the right field of view projects to the left eyeball, right? And they will travel through the respective optic nerve. But this time, the nerve from the left eye will continue to travel on the left side, whereas the nerve from the right side will cross the middle before joining the other nerve. Okay, and that goes to the thalamus and then to the occipital. Okay, so the entire right field gets projected onto the left brain, and the entire left field gets projected onto the right brain. So let's take a look at some of these questions. The right optic nerve, which is right here, the right optic nerve carries information from which field of view? So you look at the right optic nerve, right? It has both purple and blue. Right? Purple represent information from the right field, blue represent information from the left field. So the right optic nerve carries information from both FOV. Similarly, the left optic nerve carries information from both fields as well. What about the right optic tract? Okay, right optic tract. What information does it carry? Is it left? Is it right? Or is it both? Okay, let me launch a poll and see if you're all with me. Okay, the question is the right optic tract. What information does it carry? Left field, right field, both fields. Okay. So all you have to do is look at the right optic tract, which is right here. That's the right optic tract. And you can see the color is blue, right? And blue on this diagram represents left field. So that's left. For the D here, the left optic tract, right? Look at the left optic tract. It's pink, so it's going to be right field. Okay, right field. Now, next question. The right optic tract, which is the blue one again here, the right optic tract right here, carries information from which part of the eye? Okay, so you trace the blue all the way back to the eyeball. You can see it's carrying information from the right half of both eyes. And on the other hand, the left optic tract carries information from the left half of both eyes. Okay, so make sure you have this diagram handy when you're doing a test or something, so you can refer to it. All right. Let's do a poll question. Go ahead. Okay. So damage to the right optic tract will affect, and then we just have to go back to our diagram. Right optic tract is right here, and the blue one here, right? So if you damage the right optic tract, then the left field will not be processed. Okay, is that what you guys chose? Yeah, visual information from the left field of view will be affected. Okay, so I hope that makes sense. All right. Let's see. Is there any other eyeball stuff here that we should go through? Oh, we have myopia and hyperopia here. Let's quickly talk about that because some of you, well, there was that question about presbyopia, right? So let's go through this again. All right. So what happens in myopia is that we have the eyeball being very long. And this is exaggeration, okay? Normal eyeball is like this. This is normal eyeball. Okay, and this is the fovea. Okay. The lens is right here. Okay, and what happens is the light comes in, the lens will bend the light, and then it will help it focus on the fovea. Okay, so think of you know when you were in a classroom, right? There is usually a screen at the front of the class, and people would project things onto the screen, right? Either with a projector or like with the, you know, those old school ones with the transparency and all that. Okay, now without adjusting the projector, what happens if you move the screen further back, right? Okay, then the image on the screen will become out of focus. And that's essentially what's happening in myopia. All right. So in myopia, the lens is too long. The eyeball is too long. Okay. Shape of the eyeball too long. Okay. And that would cause you to have difficulty seeing far away object. Okay. So if you put the lens here, right, at the front, just like before, the problem is that the light will be coming together a little bit too soon. Okay, so light converges too soon. Right? The converging point of light rays in front of retina. Okay, the screen has been moved further back. So how do I adjust for this? Okay, let me just draw it a little bit nicer here. Okay, the lights come in. It's going to, again, this is exaggeration. Right? The converging point is in front of the retina. In other words, they are converging too soon. So to fix this problem, we want to spread out the light rays a little bit before they enter your eyeballs. And so we need to have what's called a diverging lens. Okay, type of lens is needed, diverging lens. Okay, also called a concave lens. Okay, so what the concave lens or the diverging lens does is it will take the light rays. Okay, let me draw it in pink this time. It will take the light rays and then we'll spread it out a little bit first, right? Before it enters the eye. So by spreading the light rays out a little bit, right, by the right amount, then they will now converge on the right spot. Okay, how much you need to spread them depends on how long your eyeball is. And your lens is also a little bit affected, okay? But for the most part, we can think of it in terms of the eyeball here. So if it's really, really long, then you need to spread them more and that will correct the problem. On the other hand, if you have hyperopia, your eyeball is too short. Again, this is an exaggeration. And what happens is the light ray comes in and now it is going to be converging behind the retina. Okay, so in hyperopia, you have difficulty seeing nearby objects. And the shape of the eyeball is too short. Convergence point is behind the retina. And to fix this, you want the rays to be coming together sooner rather than later in the other case. So you're gonna wear a converging lens. Okay. Converging lens. Okay. Which is also called a convex lens. An example of converging lens is a magnifying glass. Okay, so that's why people who have farsightedness, sometimes you look at them, their eyes looks a little bit bigger, right? And that's because the type of glasses that they're wearing is the same type of lens as a magnifying glass. I think that's it for the eyes, and that is 1 hour. Anybody have any question at all about anything related to eyes before we take a break? And when we come back, we're gonna do the ears. Anything at all? Okay, I'll see you in 5 minutes then. Okay, let's keep going with the ear now. Okay, let's go back to this chart here. Describe the function of the given ear structure. Okay, so first of all, we have the ossicles. Okay, so ossicles are 3 small bones located in the middle ear. Okay, so they are the malleus, incus, and stapes. Okay. And the function is to amplify sound. You can also call them the hammer, the anvil, and the stirrup. Oval window. The oval window will transfer the movements of the stay piece. So the stay piece is actually attached to the oval window into pressure inside the cochlea. So let me just show you a quick picture. I'll come back to this if you still need to copy it. So over here, right? This is your ear, where we see the malleus, the incus, and the stapes. And if we're going to zoom in over here in this section, right, you will see that the stapes presses on the oval window. Okay. If I remove the stape in the next diagram here, right, you can see the oval window clearly. And so it's kind of like a soft spot that is on this inner ear structure. So as the stape push on it, right, that will send waves, shock waves, changes in pressure throughout the fluid inside your cochlea. Okay. And then that excess pressure will need to be dissipated at the round window. So when the oval window gets pushed in, the round window will bulge out, okay, just to help alleviate that pressure. That's what the next question is, right? The next item, it is the round window. Okay, so the purpose of the round window is to relieve excess pressure in the cochlea as the stay piece, as the stape presses on, the oval window. Okay. Now, just to show it to you in another perspective, I guess, help you understand. Okay. Now, the cochlea is like all rolled up like a cinnamon roll, right? Okay, so we can unroll it to help you understand it. Okay, so unroll it, it will look like this. Tapers off into like a tip here. At the beginning here, that is going to be the oval window. Oval window. And then at the bottom, there's going to be the round window. Okay, and there's actually like a third thing in the middle. Okay, but to keep it simple, I'm just going to draw it like this. Okay, there's something in the middle here. That is where you have these special hair cells, the mechanoreceptors that we talk about. Right? They are called the organ of Corti. Organ of Corti. And so basically what happens is the stape will push on the oval window, and all these are filled with water, liquid, by the way. Okay. And then the pressure will go in, and depending on the pitch of the sound, like high pitch, low pitch, right? It's going to have different amount of energy, and they're going to turn around at different location. Okay. So that extra pressure will cause the round window to bulge out. Right? If you have a different pitch, then it might go in further before turning around and then cause the round window to bulge out. So think of the organ of Corti is located throughout this middle structure here. Okay, we're not going to worry about what it's called. And you can think of them as like piano keys. Okay, so all these keys here in the middle, right? Depending on where the wave turns around, those organ of Corti in that particular section will get stimulated. And that will send a signal to the brain, and your brain will register it as a different pitch. Okay, so a very high pitch stuff would be stimulating the hair cells that are closer to the beginning, whereas very low pitch stuff is going to be stimulating things that are closer to the end. All right. So if you roll this thing back up, you will get your cochlea. And next we have semicircular canals. The semicircular canals are responsible for detecting head movement. Okay, so detects head movement. Okay, there are 3 semicircular canal, right? Okay, so I can just draw them here for you. And this is your cochlea. You have the vestibule in the middle, and then you have 3 semicircular canal. There's one that is called the anterior. There's one that is called the lateral, and then there's one that is called the posterior. Okay, you kind of have to think of this in terms of 3D. It's kind of hard to visualize here, I suppose, but let's take a look at it in a different perspective. Okay, if you are in a room right now, right, look at the corner of the room on the floor. Right, so you have one wall, and then the floor, right? Okay, so the posterior semicircular canal is going to be parallel to this wall, parallel to the wall in front of you. You're gonna have the anterior semicircular canal that is parallel to the wall on the side, and then you're gonna have the lateral that is parallel to the floor. Okay, so this one, let me just color them for you. This here, that's the posterior one. You're gonna have one that's parallel to the floor, which is this one. And then you're also gonna have one that is parallel to the side. Okay, can you visualize that? So let me write them down for you using the same color. Let me try to use the same color. Okay, so this one that is parallel to the wall in front of us, that is the posterior one. Posterior semicircular canal. Okay, that one is stimulated when you tilt your head from side to side like that. Okay, tilting the head. Okay. This one, the green one, that's parallel to the side of the wall. That one is called the anterior one. Anterior. So it is stimulated when you nod your head. Okay, nodding the head. Okay, when you say yes. And then we have one that is parallel to the floor. That's this guy right here. That is the lateral. And it is stimulated when you are shaking the head side to side, saying no. Shaking the head. Okay, so depending on what the head movement is doing, the semicircular canal, the respective semicircular canal will be triggered. Okay. Probably better if you look at the professional drawing, which is right here. Right, you can see the 3 semicircular canal. We have the anterior one here, nodding your head. We have the posterior one, tilting your head. And then we have the lateral one, shaking your head. All right. So having said all that, let's see if you can do this one. When you're doing a pirouette, this is pirouette, those are ballet dancer. Which semicircular canal is going to be stimulated? Anyone else? All right. So the correct answer is the lateral, right? Very good. The lateral. Okay. If you go back to our picture here, right? If you are doing a pirouette, you are spinning this way, right? Okay. So that's going to be like this, spinning around in a circle. So the lateral one, the blue one, is going to be stimulated. So help me out here. What do you need to do to stimulate the anterior one? What kind of motion should we be doing to stimulate the anterior one? Anybody? Well, anterior is say yes. Okay, nothing, not saying no. But I'm talking about what kind of motion. Okay, so for example, right, this is pirouette. It's going to stimulate the lateral. If I want to stimulate the anterior one, what kind of motion the entire body has to be doing in order to stimulate the anterior one? Does anybody know? Okay, we can be doing this, right? A somersault, right? Okay, somersault. That would stimulate the anterior one. Does that make sense? Right? I don't know why it keeps on going to this page, but like, yeah, the anterior one, you will have to do a somersault. Okay. What about posterior one? What motion do we have to do? What motion do we have to do? This is somersault, okay? For anyone who don't know. Yes, cartwheels. Exactly. Let me search up a cartwheel for anyone who does not know what a cartwheel is. There we go. Cartwheel. Okay. So pirouette, lateral, somersault, anterior, cartwheel, posterior. Okay. Yeah, backflip as well, right? Yen said backflip would be the opposite of somersault, which would also stimulate the anterior one. Excellent. Okay, moving on. Next one, we have the Eustachian tube. What is the function of the Eustachian tube? Okay, the Eustachian tube is normally closed, but it will open during swallowing. Okay. Let me write that down for you. Normally closed, but will open during swallowing. Okay. It connects the inner ear, the middle ear rather, middle ear to the nasopharynx. Okay. When it opens, it helps equalize internal ear pressure with the environment. Okay. So classic example is if you're on a plane flying up, or if you're in an elevator that goes up, as you go up, the external pressure is going to drop and you feel your ear being plucked, right? And when you swallow, right, that excess pressure comes out and you feel fine again. Okay. Lastly, we have the tympanic membrane, which is just fancy word for eardrum. Eardrum. And the function is to convert sound energy into physical movement. And those movements are going to be picked up by the ossicles, which would then transfer them into pressure waves inside the cochlea. We also have the organ of Corti that we briefly mentioned earlier. Right? Contains hair cells, which is a type of mechanoreceptor. Transmit impulses to the cochlear nerve. Okay. Which then takes the information to the brain, right? If it's related to sound, it's going to be taken to the temporal lobe. Okay. That is that. Okay, I believe that is all for the ear. Any question about the ear? No questions. Okay, so I just want to finish up by going through this diagram here. Okay, so you're gonna need some colors for this. It is, you know, quite a bit of information, and we should definitely use different colors to map it out. Okay. Okay. So let's take a look here. Give me 1 second. So these are the different senses. We have the eyes, the nose, so on and so forth. So let's write down what they are called, the type of receptors, and where they're going to be projecting the information for, the information to. So this is the eye. Okay. Eye is, of course, for visual information. Okay. Now you cannot really see here because this is the surface of the brain, but we all know on the inside of the brain, you're going to have the thalamus, right? Okay, so thalamus, I'll say it's around here. Okay, so there's your thalamus. Okay, we're gonna put a label to it. Okay, so that is the thalamus, is on the inside. Okay, thalamus is going to process visual information. Sorry, it's going to process sensory information rather, and decide what goes where, right? Okay. So all of the sensory information is going to go through the thalamus except for smell. Okay. So here, the eye, visual information. So what kind of receptors do we have? I'm going to put receptors in pink. What kind of receptors do we have in the eyes? Help me out here, please. What kind of sensory receptors do we have in the eye? Anyone? Yeah, thank you, Safi and Jyoti. This is the photoreceptors. Photoreceptors. Okay, the cones and the rods. Okay, so where will the information go? Okay, so from the eyeball, the information is going to get, well, your eye is like really here, right? If you think about the location relative to the brain. So that information is going to go to the thalamus. Okay, and then from there, you know, you're going to filter out what you don't need, And the rest that you need is going to go to the occipital lobe. Occipital lobe. Make sense? So again, you should be doing this with some colors. All right, next we got the nose. And nose is for smell. Fancy word for smell is olfactory. Okay. What kind of receptors do we have in the nose? Well, scent is basically detecting chemicals in the air, right? So we would need to have a chemoreceptor. Exactly. All right, and then there are different types, right? Smell would bypass the thalamus, and it would just go directly to the temporal lobe. Okay. Next, we have tongue. Tongue. Okay, so tongue is gustatory. That means taste. Right? So taste is going to be chemoreceptors as well. Okay, but you also will be able to taste, well, not taste, but then your tongue will also be able to detect things like, you know, temperature of the food, texture of the food, and all that stuff. So there is going to be some mechanoreceptors, maybe, as well. Okay, and some thermoreceptors. Okay, and you know, if you bit your tongue, it's gonna hurt, and so you will have some nociceptors as well. Okay, tongue is basically, you know, covered with skin, if you think about it, right? So everything you found in the skin, you will also find in tongue. Now, this part is the part that might get a little bit of confusion. So see if you understand. Right, so over here, the tongue is gonna go, you know, through the midbrain, okay, and then it's gonna go up to the thalamus. Okay. So the aspect that relates to temperature, texture, and pain, all that stuff is actually going to go up to over here. Okay, that part is going to run out of color soon, but that part is basically the primary somatosensory cortex, right? Yeah, I'll label that for you. This is primary somatosensory cortex. Okay, that goes right there. And the taste aspect, right, whether something is sweet, sour, bitter, that will go into the insula. And you cannot really see the insula here, but then it's, if you pry open the frontal lobe and the temporal lobe, it's going to be right inside there, right? There's the fifth lobe, right? Okay. So there is that. Okay, next we have like the skin, right? Basically. Okay, and these are somatosensory information. Okay, so like the pain, the pressure, all those stuff. So of course we have the mechanoreceptors, basically all these things, right? Okay, I'm gonna maybe copy it here. Copy it. I'm just gonna put it right here. Right there. It's the same stuff. Okay. No chemoreceptors though. We cannot taste with our skin, right? So that information, let's do orange. It's going to travel up the brainstem, gets to the thalamus, and then it's going to go project to the primary somatosensory cortex. Okay. Next, we have two different things here. Okay, so we have the ear coming up, so I'm just going to be focusing on this part here. Okay, so this is the vestibule, right, plus the semicircular canals. So the receptors we have is mechanoreceptor. Okay. And the vestibule is for acceleration. So, e.g., speeding up or slowing down in a car, right? Falling, right? And so when you're on your roller coaster, your vestibule is very active, right? Detecting the falling, going down, going up. Okay. Whereas the semicircular canal is for head movement, right? We talked about that already. And so all these information is related to, if I have to put it into a sense, it will be like equilibrium. Okay, equilibrium. So where does that go? Anybody remember? Information related to equilibrium, where does it go? Help me here, please. Where does equilibrium information go in the entire brain? Where does it go? Mary says cerebellum. That is correct. Very good. So it's going to go right here. Okay, almost balanced information, so on and so forth, right? Okay. Lastly, we have the ear. We've got the ear, which is right here, ear. And that is for auditory. Okay, auditory. What kind of receptors will there be? What kind of receptors will be in the ear for the cochlea? Okay. Let's see. Cochlea contains mechanoreceptors as well. Okay. Yeah. Thank you, Safi. And I think I'm out of color. Let's just do, which one have we not got yet? Let's do hot pink right here. That is going to go up here to the thalamus, and then auditory information gets processed in the temporal lobe as well. Okay, so this diagram is a little bit messy, but it's an important one because it helps you map out where everything goes. Right? So I encourage you to maybe draw one on your own when you have time. Any questions? Okay. Let's take a look at this application question. You stare at a bright red screen for 30 seconds. What color will you see if the screen switched to white? Anybody know the answer? What color will you see? Red? Okay, anyone else? I'm trying to find the color wheel to help you understand. There's the color wheel that we have in the lecture notes. And so if you stare at a red screen for 30 seconds, what happens when you switch to a white screen? Okay, why don't we try that? Okay, why don't we try that? Let me create a bright red screen for us to stare at. That sounds fun, doesn't it? Bright red screen. All right, so here we go. Let me switch the screen. Okay, there is your bright red screen. So why don't we stare at that? Okay, don't even blink. We're gonna do it for 30 seconds. Now, in order to see this red, right, we have three types of cone cells, where we have the red cone cells, the blue cone cells, and the green cone cells. When you are staring at this red screen, the red cone cells are the ones that are working really hard to register the color. Okay, so all these red cone cells are sending signals through the optic nerve, which then goes to the thalamus and then to your occipital lobe, and you register that as the color red. Okay. Now, if you have been staring at this screen for the past, whatever, 30 seconds that I was talking, now I'm going to switch to a white screen. I want you to blink a couple times. Okay. What color do you see? What color do you see? Okay. So let me go back to the color wheel here. Now, a lot of people say blue. Now, did you see this blue, or did you see like the cyan? Yeah, the cyan is probably more accurate description. Not the dark blue, right? But the kind of like the, yeah, the cyan. This is called cyan. Okay, so here is why. In order for you to see white, okay, in order for you to see white, you need the red plus the blue plus the green. All three of them has to be firing at the same intensity for you to register the color white. Okay. However, when you were looking at the red screen, the red cone cells were working so hard. The blue and the green were doing nothing, right? On a red screen. It was just the red that's working like super hard. So when you switch to the white screen, right, because the red was overworked on the previous slide, this one is like not really working for the moment. It's like resetting itself, it's taking a break. So the blue and the green are the ones that are firing really, really strong on a white screen, but not the red. As a result, you see the combination of blue and green, which is why it's cyan. Do you understand? Okay, thank you. All right. Well, what if you switch to a green screen? If we stare at a red screen for 30 seconds and then we switch to a green screen, what color will we see? What color will we see? Will we see red? Will we see yellow? Will we see green? Or will we see magenta? Okay, let's do a poll. Okay, here's the question, right? You stare at the red screen for 30 seconds and you switch to a green screen. What color will you register? Okay, anyone else before I show you the answer? Okay. All right, let's think about this. How do you see the color green? What do you need? Which two receptors do you need to see the green color? or which receptor do you need to see the color green, I should say? Receptor or receptors? What do you need to see green? To see green, we need green cones, right? There are only 3 types of cones: red cones, blue cones, and green cones. You have to understand that. So to see the color green, you need the green cones. So if you were staring at the red screen for 30 seconds, the red one gets exhausted. But when you switch to a green screen, you didn't even need the red to see the color green, right? So it doesn't matter that the reds are exhausted, okay? To see green, you only need to have the green receptors. So you will have no problem seeing the green. So the correct answer is green. Okay. However, if I switch to a yellow screen, right? Yellow is red plus green. Okay, red plus green. So if you exhausted the red by staring at a red screen, when you switch to a yellow screen, now it's going to be appearing as green. Okay. Are we together on this concept? You're probably going to see like 1 or 2 questions on this on the test. All right. Okay. Next application question. David went for his yearly eye examination and was informed that his intraocular pressure, that means the eye pressure, right, was slightly elevated. The physician expressed concern over this condition. Note that if the condition got worse, eye drops would be needed. What is wrong with David's eyes, and what are the possible consequences of this condition? We talked about this already, right? If the eye pressure keeps on going higher and higher, right? If the aqueous humor, the vitreous humor rather, it's putting too much pressure on the optic nerve, that could damage the optic nerve over time, and that could lead to glaucoma, right? So if you give him eye drops, the function of the eye drops would be aimed at relieving the pressure of the eye. You cannot reverse it, but you can slow the progression. Okay, there's no cure for glaucoma as of yet, but you can slow the progression of the condition. Okay, my brother-in-law has glaucoma, and he needs to regularly put drops in, and in fact, one of his eye is much worse than the other, so he cannot really see very well with the affected eye. So there you have it. That is the application questions, and that is it. Okay, for today's tutorial, I will, as usual, post the answer for the fill in the blanks, and you can check that up yourself. All right. Thank you for coming. I will see you all next week for the lesson 8. But just hold on a second. Let me see what's going on next week. Just to be sure. I think we also have the lab coming up, right? So let me just pull up the calendar for a second so we know exactly what's going on. Calendar right here. Okay, so next week we'll do lecture 8 plus lab 2 take up and test 2 review. Okay, so I will put the link on eCentennial so that you would have access to the test 2 review and stuff like that. And you obviously have to do the lab 2 first before coming to the tutorial. So it's very busy next class. Probably we will have to go the full 3 hours. But yeah, we'll see how it goes. Okay, thank you. Bye for now.